Right. So actually, there are three themes that I wanted to explore, and actually, we'd almost think this was planned. We've got the three themes covered with the with the three people here. One was to look at actually the technological innovations that are actually coming in the pipeline, and I think Claudia, you can speak to that in particular. Then there's the linkage of those with the data infrastructure that we need to actually make those speak and allow us to understand what they mean and how they direct to different treatments, etc. I think you can speak to that, Jürgi. And then also how we translate innovations to the actual healthcare setting and ensure that we get equitable and better outcomes from them. And that's where you come in, Hugh. So Claudia, let's ask you first about the technological innovations that can improve early diagnosis and early diagnosis.
Well, what a big topic to start with. So maybe let me take a step back. I guess what we've seen, precision medicine has been around for, 20 years at this point, and I think we're now coming to an age where it's truly becoming precision medicine. So we coming away from the paradigm of having one diagnostic for one drug, one very specific pairing, to having now combinations of therapies with different biological ways of acting. We have a lot of the AIs coming into play, as well as more advanced technologies. So I think all of that together is really forming an ecosystem that allows for much more in-depth assessment of the patient status and for much more directed treatment. So we're at a position now where we can truly identify the appropriate treatment for the appropriate patient on a much more in-depth basis than we were able to understand previously.
Mm-hmm. And in terms of so that would be sort of identifying and characterizing the patient. Then we've got things like liquid biopsies and things which allow us to make for an easier or quicker diagnosis as well and characterization.
So yeah, that's one of the advantages is liquid biopsy. We've already heard a number of references to liquid biopsy. So maybe let me explain for those that don't know what that is, explain a little bit what that is about and why we're so excited about it. So liquid biopsy, as the name implies, is based on the liquid specimen, in this case blood, that allows for easy access to the specimen. So, I guess the other thing is liquid biopsy testing assesses typically tumor that's shed by tumor DNA. So DNA that's actually shed by tumors as the cancer cells degrade. So what that allows us to do is actually see the tumor in a blood specimen. And why it is very exciting, because a blood draw is obviously very attainable. So it spares patients from actually having to do more invasive procedures to obtain a specimen from them to be able to diagnose. So since a blood sample is so attainable, we can now no longer just diagnose at the early, at the time of surgery or after one particular biopsy event, but it allows for more frequent testing. So it allows us to monitor patients to see if there's recurrence in disease. And I think we had a patient talk about this very neat a little bit ago, like an hour ago. So it allows us to actually use the testing in very different ways. So either for diagnosis, for monitoring of disease, for recurrence, or also to make treatment outcome predictions. So it's very exciting technology. And again, there's a lot of benefit for patients.
And also it speaks to the evolution of resistance to particular treatments. So you need an easy method to keep monitoring the treatment as keep monitoring the response to treatment as the treatment progresses. Yeah,
exactly.
So Jürgi, I mean, we often this personalized approach to diagnosis and then the treatment decision making. People speak almost. It then trips on to data and AI. Why? Maybe you can explain why and why that's important and can make that connection, because people often just bring them together.
Sure. Well. Yeah, actually, our company was funded in 2011, and our tagline is democratize data-driven medicine. Right? Mm-hmm. So we would see precision medicine already beyond objectivizing the outcomes and the decisions around data, which I think is well needed. But indeed, the technologies that Claudia was talking about, so for example, for liquid biopsy, right, are digital technologies. So this is next generation DNA sequencing, which has been enabling, I think, a lot of discoveries in cancer over the last 20 years. But now those devices have been miniaturized and are available as well in hospitals, right? And so They enable better testing of patient cancer cases in hospitals. We work with, I think, 350 hospitals ourselves in Europe who have these capabilities of producing this digital data. And thanks to this digital data, then one can understand the cause of a cancer. Because there as well, over the last 20 years, I'm a molecular biologist, so for me it's been fascinating as well to follow that. We've discovered that actually cancer is a disease of DNA, right? And what one needs to look at is DNA or genome integrity and DNA damage repair systems and so on. And so now you have these fantastic technologies that enable you to understand the origin of a cancer or of how cancer has evolved, in particular looking at the content of the mutations in the genomic profile of a patient, which has been digitalized. And what you have to do is make sense out of it, right? And these are very complex data, so you need smart algorithms. And whenever you have smart and complex things, it's as well very important to have a lot of diversity of data, because the more diverse data you've been facing, the better algorithms you will be able to develop to solve more cases, right? And so hence the importance, if you like, of data, the importance of cloud, the importance of algorithms in that space. For us, the ultimate end is to have our platform deployed in as many hospitals as possible. Today in Europe, we are in Gustave Roussy. Here in Brussels, we are at UCL. In New York, we are now at Memorial Sloan Kettering, so that the more patient cases you get in a single platform, the more data, the more you can learn, the better you can compute the data, and the better you can help the oncologist, the pathologist, of knowing how to treat the case of one patient on the basis of similar cancer cases that are being already computed in a single platform.
So that presumably that assumes, I suppose, that you're not only collecting the sort of digitized genomic information from the patients, but you have to link it to what then happens to them in the health system as well, so that you can then correlate.
That's super important. So if we take and we make a very simple version of cancer, right? To really understand cancer, you need to have the molecular profile, because this is what is driving cancer. Otherwise, like fixing a car without having a proper diagnosis of the car, not knowing what is broken in the car and in the engine in particular, right? And then to your point, you need to have a longitudinal perspective of the patient. So this can be obtained in some cancer cases by liquid biopsy, but primarily today is CT scan, PET scan, MRI data. And now when you have the picture, the video with the treatment or the sequence of treatment that patients have received, and then the outcome, you can even start to cohort patients and eventually predict that for a given patient that has a specific cancer, the best path is to start with treatment A and then go to treatment B.
Right. And so just to clarify, is that a research activity, or are you using it then to, if I come in, am I contributing to the body of knowledge that will increase understanding in 10 years' time, or does this directly feed back and affect what I will receive or what happens to me?
So to give you, I would say, very practical numbers, we are, I think, one of the dominating players in that space in the world. And so our data are so that we have already been computing 1.4 million genomics profiles that are being produced by hospitals, right? So that's already real. Now, when it comes to combine this phenotypic data, longitudinal data with genomic data, today, this requires to have prospective studies, right? To demonstrate that you have a value and that you can predict to have response. So in our case, today, these tools are for research use only, but we've demonstrated, for example, in non-small cell lung cancer, stage 4, that by following over 2,000 patients now on 200 clinical data points, we can predict response to immunotherapy with 80% AUC.
Right. So, I mean, presumably these kinds of relationships are going to be critical for your business as well, in terms of ensuring that the right medicines get to the right patients, and that you're maybe discovering, working out what the next mutation that you want to target is, and so on.
Yeah, absolutely. And so we had a lot of discussions earlier already that touched on this. So this would fall into the, anything that's used to guide clinical, any technical tools, healthcare tools they use to guide clinical decision-making of regulated products, right? And so there's an ask from regulators to obtain certain confidence in a technical performance, right? And a technical reliability, as well as a clinical performance. So I think that's a symbiosis there between pharma and the health tech fields, right? In terms of leveraging each other's strengths, generation of clinical outcomes data, and the technical abilities. So yeah, absolutely.
So Hugh, to you now about the translational part. So this is all sort of cutting edge, the sort of marriage of bioscience and computer science, for want of a better term. How does that translate into the health systems, and how do we make sure that innovation which is that gets in effectively but also safely, because it's new technology, so it has the risk of doing harm too?
Yeah, so it's a very good question, and maybe I could just set why I'm here in
Yes,
context.
yeah, that would be yes, maybe a good start. Yeah.
No, it's simply because I'm not a cancer specialist. I don't work on cancer. I work at a public-private partnership between the European Union and 5 different trade associations covering medtech, pharmaceuticals, biotech. And we have our goal as funding projects, so we don't actually do any work ourselves. We're trying to fund projects that are going to facilitate the translation of research and innovation into benefits for patients. Because when you come to conferences like this, it's very easy to get very excited by the advances. However, a diagnosis is not a diagnosis. It's not a helpful thing if you don't have an appropriate treatment, if you can't link that appropriate treatment with a regulatory framework, if you don't have a system that then may allow you to reimburse that. So all along, if we want to bring innovations to patients, we have to find ways of linking all these different pieces together. And if any of you have ever moved from city to city, or changed clinician, or changed your hospital, how many of you know where your health data is? I think most of my health data sits in Ireland. I don't know, I've never needed to go to hospital in Brussels. So how are we going to make those links, and how are we going to put everything together? So that, in a very general, fluffy way, is what we're trying to do. And we're trying to build consortia of public and private partners. So these are patient organizations, regulatory bodies, researchers, companies big and small, hospitals, and actually getting everybody together. And we have 2 challenges. First is, what is the challenge? Can we actually agree on what is the blockage that's preventing an innovation from getting to market, from actually getting to benefit patients? And then, when we've actually identified what those challenges are, do we have the capacity to build a data set that gives the decision makers the evidence they need to either decide that it works, that it's appropriate in a certain population, that it should get regulatory approval? So I apologize for that long introduction,
it's
but
very helpful to have that as a start.
it's just important to explain why we need platforms such as IHI. I'm biased. I'm the executive director of IHI, so I'm going to tell you that. But when you're saying about, well, okay, how are we going to bring these innovations to patients? Well, these are the questions we really have to tackle. We have funded projects in the cancer space. So between IMI1 and IMI2 and IHI, I think we have 16 projects tackling different things. We've worked on liquid biopsy, but we've done really boring stuff. It isn't the exciting research cancer bit. It's what are the protocols you need to run when you're collecting liquid biopsies? What's the data you need to capture? What are the contexts, and how do you have to adapt your protocols depending on the setting? Because this is the foundation upon which a lot of the innovation will then be able to reach patients. And we can't, I'm sorry to be so boring, but we can't really underestimate the importance of this. If you have lots of companies, lots of hospitals generating lots of data, to actually make use of it, to actually get the evidence that we need to make decisions, you have to be able to link it together. So we need to put a lot of effort into understanding problems, standardization, and harmonization. And then that is the ground on which innovation can really flourish.
Okay. Any questions from the floor? Otherwise, I'm going to... Yes, please. Just coming to you.
Thanks. I was really interested to hear that last point. Oliver Kemp from Prostate Cancer Research, and yesterday we were talking about trying to get approval for new diagnostics to use in prostate cancer. As you may be aware, some of the diagnostics for prostate cancer are old and not very effective. Yesterday when I was talking to the Health Secretary in the UK, he was saying exactly all the things that Hugh was just saying. If you're going to get these things approved, you need to make sure you've got all the data, it stacks up, and we cannot approve anything to replace the PSA or to implement universal screening programs until we've got the data that shows that it's that we're not increasing the risk of over-treating people. And I noticed the EU's already said we're going to do PSA plus MRI for the whole of Europe. But the UK's got a harder line on that and is scared about the risk of over-treating. But I'm really happy to hear Hugh say it's the boring stuff that matters, because actually it's 12,000 lives that are at stake. Yeah. 7 or 8,000 of which are completely savable by us making sure that we have the right screening programs in place.
Mm. So I just, maybe if you don't know, I direct you to a project from IMI1 called Pioneer in prostate cancer. And they're looking at the data and trying to use the data that actually exists, that's coming from different hospitals, different clinical settings, and trying to compile that data in order to be able to look at, are there ways to improve screening, to improve diagnosis. It's only a small step, but these are the sorts of projects that we are trying to support, because if we can provide some evidence in one area, then we're hoping that we'll have the knock-on effect, because then others will begin to step into the field and add more data.
But Hugh, an extension of that, it's not simply generating the evidence, but making sure that you capture how you do that. So if it's something like liquid biopsy, who does it? What's the lab standards that are used? Yeah. How is the information then fed back into the multidisciplinary teams? That kind of thing. So I guess it's because if you're going to show effect, you need an optimized system, and then that needs to be transplanted and disseminated.
Can I intervene?
Yeah.
Because for us, it's our day-to-day, what we're talking about. And in liquid biopsy testing, we have a lot of hospitals that are working with us, different ways of producing data. But the way you harmonize is post-data production, right? And that's why algorithms are so important. And I would like as well to set a bit the standards and the stage here. In an hospital today in Europe, no one goes live with a new test unless it has been compared versus a legacy technology. Yeah? So just to break any myth, right? So hospitals are very responsible when they go live with a solution. They need to bring a solution that is as good as what they had in the past with some additional benefits, right? So I think the word that we are hearing and describing for me already exists. This is what we already do ourselves. So we could tell you, for example, which are the labs that are doing proper genomic testing, liquid biopsy testing, which are the patients that indeed are being or not being tested on prostate cancer, eventually as well, knowing already ahead of time among those patients, which are the ones better responding to specific type of treatments because of specific molecular signatures. I think this word of real-world, real-time epidemiology, this is how we would call it, already exists. I just think in the context of what you were describing that we need to advocate a little bit more for industrialized solutions, because if we don't break data silos across one common platform, two common platforms, we will never create this collective intelligence. And I think this is, I've been living 10 months now in the US. This is where in Europe, unfortunately, and I was telling you I'm super pro-European, but we have to be more federated. We have to be more together. I think we have a huge disadvantages sometimes versus the US, which is one market. And I think I would like a stronger regulatory body in Europe that is imposing countries as well the fact that we need to move more quickly at scale with industrial solutions in those considerations versus remain on the research side, because we need indeed to impact on patients.
Mm-hmm. Yeah. But I think one of the things, and I don't disagree with what you're saying, that advances are being made, but how do we get to the point of having this level of standardization for all tests, for all cancers, across all hospitals, And the thing is, we're not there yet. And we have to find this way of collaborating, of working together. And I think we pull a lot of the strands of the different conversations that we've heard today. You need the regulators, you need the patients involved. And there's a great opportunity there to build a lot of what we need is we don't need lots more innovation. We actually need to understand what we've got, because there's huge amounts of data that are lying unused, and we can tap into that, and then based upon that, you then have that fertile ground.
Maybe to add one
Yes.
thing, so as a former product reviewer, I guess I'm very passionate about the whole technical development topic and standardization. I guess standardization allows us for a number of things, and I think we're talking about really complex technologies here, and differences in design can really lead to different technical outcomes. The issue is if you have a complex clinical outcome landscape too, then it gets really difficult to connect something that's variable on both sides. And so standardization really is a wonderfully powerful tool that we as a company also for sure participate in, because that allows you to then amplify the power of the clinical evidence generation. So how do we actually then use those more standardized tools to make predictions for patients' well-being of reoccurrence, right? That's how you achieve those, by standardization of technology. And that allows you for, I guess, assessments, any AI-based assessments and conclusions drawn from a particular data set are facilitated by having standardized inputs.
Right.
I agree, but may I give a concrete example? PARP inhibitors, you have PARP inhibitors as well. Okay, so we have helped a lot of hospitals identify an HRD status, genomic instability scores. The data are excellent, actually excellent even versus the compound, irrespectively of how the data are being produced. Today, there are not enough hospitals that are being set up with these capabilities. Why? Because actually the reimbursement, the testing is not generous enough. And so again, I go back to my say on, I think you have to have the right level of funding. It never has to be too big, because otherwise we do bad things as well. But in Belgium, top of my head, testing for solid tumor with NGS is reimbursed 500 euro. In the US, it would be 3,000. 3,000 is too expensive, but should be 1,000. So what I'm telling is that, as well, they're being innovative on how we can directly impact through this type of things, and I don't know whether this is part of as well IHI, but this is, I think, what we need, because to your point, technology is there. There are cases like prostate cancer where you have two choices. Either you do new liquid biopsy test, or you go back to a block which is 10 years old. I mean, your cancer has evolved. What's the point of testing a block which is 10 years old to decide what therapy should be given to a patient? So I think the technology is there, the industrial solutions are there. What we need is support.
And maybe if I could just come back on that. I mean, it's very interesting. I mean, we as a program are funding, we're doing a bit of a John the Baptist, so we're trying to make the path straighter. Our job is not to put innovations onto the market. There are other parts of the European Union. So, but then if you think of the regulatory, you think of reimbursement, that's a huge part of which is outside of the scope of our program. But it's a huge part
Yeah, it's
of
really critical, yeah.
And it is as important. But I come back to the point. We have to do everything we can to generate the evidence that shows this should be reimbursed. This is how it should, this is the impact, this is the quality you're going to bring to the patients. And to convince the regulators before that. For me, this is where the working together is needed, because we no longer can just work on our one little piece at different parts. And actually, when we talk to some of our projects, even though we're not designed and we're not set up to really change regulation, we try to generate data that might inform regulatory science. We're not about changing regulation, but we try to get researchers to really think about these questions right at the beginning when they're designing their studies and they're designing, so that actually some of this is built into the way we approach the problem in the first place.
Well, to deliver value to the patient or to the health system,
Yeah.
it's only as good as its weakest link along that pathway. You can have a great diagnostic, a great therapy, but if it's not being delivered within an optimized system, you're going to end up, yes. Sorry, question here.
Yes, hello. My name is Geert Martens. I'm a medical doctor and head of one of the NGS labs in Belgium. So
Right.
as Jurgi was discussing, my lab faces many difficulties. We have fantastic technology. We have comprehensive genomic panels, soon whole genome sequencing, but there is no reimbursement for this. So basically what we do is that we finance this testing for the patients with a, let's say, a total cost of ownership of around 1,500 euro for a large panel out of our own pockets. And we finance that with the profits that come from other parts of the lab, since we are an integrated lab.
Right.
Today in Belgium, for instance, we have guidelines for brain tumors that recommend certain testing, methylation profiling, but there is no reimbursement. So the guidelines tells us you should do it, and we have no money. And for me, one problem today is that there is a silo between the budget for testing and the budget for the therapy. And so we know the concept of companion diagnostics. So I often tell my government administrators and the politicians, you should break up that boundary. And when a new precision drug comes to market, and that costs, let's say, 40,000 euros per year, we'll not discuss about that high cost, then let's say a yearly budget of 3,000 euro for one comprehensive panel and one comprehensive liquid biopsy, it's really a rational choice. It's not an excess. So this is one way, let's say, to create value-based healthcare by joining testing and drug budgets. And the second thing that I heard today also in the panel, even though I don't like monopolies and, let's say, standardized or enforced solutions, I think there is no other way. For the testing, either we should all go maybe in a couple of years to standard whole exome or whole genome testing of tumors. But I think consolidation on really large panels, I'm talking like TSO 500 or the Roche FMI solution, that will be the way to go for two reasons. First of all, this allows labs to consolidate multiple flows of samples from multiple tumors in a tumor-agnostic approach onto one solution, which is much more cost-effective, allows us to centralize the sequencing on bigger sequencers so that it becomes less expensive. And second, this offers a future-proof solution also for the industry that they know with which test the patient was tested, and it allows data pooling.
There's a lot in there. I wonder, I mean, one thing I want to pick out, well, for one thing, in some countries, there's the concept of a bundled tariff for the whole pathway, where you negotiate on the basis of innovations along the pathway, then you kind of negotiate and one can argue about whether that's enough or not. That's one way of tackling it. I also, you raised the issue of companion diagnostic as a companion diagnostic, and therefore the bill for the diagnostic is such a small fraction of the. But is that actually ultimately detrimental to the argument? Is the tension between that and just seeing it as, because if we don't know what it will reveal, we don't know that it might lead to treatments in the future, development of new treatments, it might use repurposing of old medicines. So the concept of a companion diagnostic and using that as an argument may be actually detrimental to the idea that we need whole genome or large-scale sequencing because it's an investigative modality whose consequences we can't tell, but they're bound to be good.
Yeah, I get your point, but if we had, for each cancer patient from, let's say, beyond stage 2, if we had access to a comprehensive panel, that gives us a lot of information about point mutations, fusions, MSI, HRD status, and whatever.
Right.
Then we get, let's say, the first diagnostic snapshot, and then later on during the evolution, this kind of testing, whatever the solution involved, would allow for umbrella or basket trials, etc. If those data can then be aggregated in a national tumor board,
Yes.
this would give a very interesting access for pharma players to get their drugs at the right
Yes.
patients. But the problem today is that the budget for this test does not become available, and this slows down innovation, or at least the translation.
I mean, the previous presentation was about equity, right? And so not having, in my opinion, any regional hospital, I don't know, maybe 10 in Belgium, maybe 14 in France, close to patients who can do that, is preventing equity as well. And to your point, and we go back to reimbursement issues, and thank you for highlighting that, because I know that in particular in Belgium it's very difficult. This is why, and Claudia knows that, you have medical affairs as well in pharma, because their worry is as well equal access to compounds for patients. So yes, there is a challenge in Europe. Again, I'm sorry to say the technology is there. We can debate, show whatever you want. We ourselves only work with labs which are BILAC in Belgium, COFRAC in France, and so on. The problem is elsewhere. The problem for me is in having the adoption of these technologies into hospitals, because indeed they can afford that, and we had a nice testament
So
here.
I think my hospital, my lab did around 500 comprehensive liquid biopsies with the Roche Aveiro technology. So the total cost is around 1,400 euro per data point. Half of this was funded by research grants. 3 were built to patients, rich people. 2 cases were paid by the insurance. But this is just because the insurance didn't know what they were paying for, and all the rest was paid by my own lab, because we just, don't want to say no, and we want to be,
And then, if I may, another challenge is not having enough experts. So when we think about innovation and so on, this is a new medicine, having experts who are capable of doing interpretation of those cases as well.
Yeah.
And maybe, again, I shouldn't make publicity, but for instance, the choice that Roche made, they purchased Foundation Medicine, put a sequencing lab, a central lab in Germany. And so this is a major player in oncology treatments. So they produce and they make most of their profits as a company with their therapeutic arm. Then there is a second branch, which is the diagnostics. And so they kind of insourced this comprehensive genomic testing. And I know that a lot of clinicians are just using it within clinical trials, but even without clinical trials. So this is something I understand from the company perspective, but on the other hand, as a medical doctor, I think it's very important that there remain independent molecular pathologists, clinical pathologists who run those tests. And so, but this, I think, should be on a consolidated solution.
So when we're talking about this relatively new technologies that are very dynamic as well, they're evolving fast, of genomic testing and its application to disease, not just cancer. I mean, I feel that we've kind of been here before, and some of you may have heard me say this before, but we've kind of been here before in the sense that, 100 years ago we discovered X-rays, and radiology evolved as a community of practice with standards, with medical physicists. A whole thing arose around it, which is independent of the application. It was never seen as a companion diagnostic for it might have been at one stage for a fracture, but it now underpins everything we do in pretty much everything we do in healthcare. And I can't help but think that particularly genomics, not only in cancer, but in autoimmune diseases and maybe even in infectious diseases, well, in COVID we were sequencing. So there is a need to develop this as an infrastructural element to our health systems. And somehow we've been there before. I don't know what I'm trying to get at is how are there lessons to be learned from that past? I know we were in things were cheaper then, and it was a different whole different era. But are there lessons we can learn from that in thinking about it today, instead of thinking of it as a companion diagnostic, or thinking of genomic testing as having a particular application, but actually seeing it as an infrastructural
Well, I think you cannot generalize the results of genomic testing to that extent at this point. You're
Not at this
comparing it to-
point.
yeah, so you're comparing it to structured data like
Yeah.
X-rays or imaging results, right?
Yeah.
That are very, I guess, very standardized images. But,
Yeah.
but that's where Hugh comes in.
Yes.
No pressure. But no, that's where standardization is really important, right?
Yeah.
So that we can actually start to generalize observations, right,
Mm.
and clinical outcomes associated with them.
And I also think in certain areas the genetic links, the genome sequencing works brilliantly. Others we don't know.
We don't know.
And
No.
the problem then, it becomes more of a societal and ethical one.
Of course.
Are you then going to start sequencing and give people genomes where we aren't sure when it comes to mental health, when it comes to...
I guess that's where biobanks come in, don't they?
Yeah,
We're kind of
but-
extending beyond our remit for
Yeah,
this session. Yeah.
but I think there's quite a big step between
yes.
the discussion where we know some things about specific cancers
Mm.
and specific mutations, to then this sort of seeing genomics and genome sequencing as some sort of infrastructure that you can go in and you know, okay, I'm probably at 50, so I probably need this sort of treatment.
Right.
Because one of the things I'm very interested in is pharmacogenetics.
Mm.
And if you wanted to reduce the number of adverse events, we already know hypersensitivities, we already know mutations for a range of different things. And, you know, the question, it's a completely different question. Well,
Right.
it's not a completely different question, but it expanded maybe beyond
Yeah,
our
okay.
scope into, well, how do we then get these tests, you know, into primary care? Because that's where you would need them, or into the A&E before you start treating people.
All right. Well, let's reel it back from there
Yeah.
because it's a bit far for us now for this session. But I do wonder whether
But
that
you asked
broader-
learnings.
Yeah, because
Can
that's
I
what you're
have
doing.
one?
You're talking about learning.
Can I have
from-
one?
Yeah.
So I think the big difference is on anything you said, there was no cloud. There was no ability of creating a collective intelligence. This is a new paradigm. And again, when I refer to the real-time, real-world epidemiology, I think this is what we have to create. You know, I will give you an answer on COVID. I was very frustrated. We are plug and play to over 350 sequencers in Europe, okay? We tried to reach out to the European Commission. In 3 months' time, we developed an algorithm to be able to have COVID surveillance. We already plug and play to the sequencers, okay? These sequencers that were already sequencing the COVID virus, if the data would have gone to our platform, we immediately had a view, real-time, real-world, of how the COVID was evolving. So again, technology is there. I agree with you that there are some points where it doesn't make sense, but let's make the difference on things where it makes sense.
So you did mention reimbursement. So is that completely outside the? I mean, as an important, but that's not within your remit to talk.
So what we are trying to do is to build, you know, the collaboration around the challenges, so that we have the evidence that either means we agree with the challenges, which is sometimes more important, because if you have everybody thinking we're working on this, but they're actually working on different things, we don't stand a chance of making progress. So get agreement and understanding some of the problems. And, you know, we have 2.4 billion euros. 50% comes from the EU, 50% comes from our members, our industry members, spread across 7 years. So compare that to the budget of the US for healthcare. Compare that to the budget of Germany, to France. It's a relatively small amount. And so therefore, we have to pick where we need to work, and we kind of make incremental changes. And reimbursement is a political question. It's a political decision. And we try to focus on informing, generating scientific evidence or regulatory science data that allows us to make better decisions. And I'm afraid I'm not the right person to talk about reimbursement.
Claudia, what's the situation? I mean, in the US, with this sort of innovation, if you know the answer to that, but with reimbursement of companion diagnostics or of NGS for, I mean, is it widespread amongst insurers? Is it mandated federally, or how does it?
As far as I'm aware, it is automatic reimbursement by Medicare. Thank you.
Okay.
For companion diagnostics that are approved by the FDA.
Right. Okay. Right. And that's not the situation. I mean, we don't have that situation in any
No.
countries in Europe.
No, every country different. France, you receive a budget. Let's say you think you're already doing, and we're talking about patients you're already doing. So utility is clear. So you're doing 1,000 patients a year. They will tell you, we give you a budget for 800. And you may end up doing 1,200 and find money elsewhere, bring efficiency, which is never bad. That's why I'm not pro to have too much money, but it has to be the right level of money to be able to do the delivery. Germany, you're being reimbursed on a case by case. There are being some abuse, by the way, a long time ago. And Belgium, I don't know, but I know that is very bad.
But maybe
It's one of the most complex systems. It's like Egyptian hieroglyphs.
nice. If I can add one more thing to this, though, just keep in mind that companion diagnostic in Europe are a relatively new concept. So we have the EU IVD regulations is coming into force last year. So the very notion of, the very definition of companion diagnostic, and the very first companion diagnostic to actually be approved under the new regulation, that's actually a relatively new experience, a very new experience. So this is actually a good time to bring this up. So if you want to bring this up, it's the perfect time to bring this up because we're actually making that experience for the very first time right now.
And it's also a reason why we need health economists. Because we need to be able to make the link through. You know, it's research, it's everybody. We need a really good understanding of what are the benefits that this can bring, and then health economists can then take on the policymakers and provide.
But isn't it a bit of chicken and egg? Because no one's going to fund it, or you're funding it out of, we're doing little bits and pieces. How do you generate sufficient evidence? Or maybe you accept it from the US or from places where it's already established. How do you break that?
Well,
I need evidence, but I can't fund it, so you can't make evidence, create evidence. I mean...
But some of the evidence already there.
Yeah.
Some of the things are set in practice, So let's see what, I don't know, we may have met already, but what he's doing and what is the benefit. And let's do that in 50 centers where it already works and really see the benefits and try to, then make the learnings and deploy that to other centers with good practices.
And I think we have many examples from COVID. We have a project called Eden, which is this federated databases across Europe about compiling data, about standardization of data. They didn't have to do any new studies when COVID happened. When a certain American president said we all should be taking hydroxychloroquine, as a treatment, all they had to do was look at the available data, look at the clinical studies, and they were able to show that it wasn't safe.
So that was coming out of real-world practice?
That was data that had existed before.
But
COVID.
from real-world practice
From
rather
real-world
than...
practice. They just went and they looked for the... So I think we have to do... That's what what I was saying at the beginning. Sometimes the solutions are not to be more innovative or to do lots of new shiny things. Sometimes we have to look at the data we have and use it better.
So we're happy to help. If you want data, we probably compute 200,000 genomics profile this year in Europe across 320 hospitals.
We've just
happy to share that with you.
So we've just launched a project called Moderator, which is about using existing data to try to improve lung cancer diagnosis.
Okay.
We should discuss.
Yeah, we should discuss.
We've got a couple of minutes left because there's reception afterwards. But so it's up to you when you want to keep going and miss out. But go ahead.
Well, just from a patient perspective, I was diagnosed in 2015 and I had 2 biomarkers in my diagnosis that were tested for. My genetic information came about 18 months later, which showed that I wasn't Lynch syndrome, which sort of was useful. However, they also said it was inherited. There was an inherited element. And we only have tests for FATMAP and Lynch syndrome in CRC. We don't have the other ones. People come to me today to ask, say, I've just had a colonoscopy, I've got a tumour, what can I do? And some of these are quite wealthy. In fact, if I was at that situation now, what I would do is I would go and pay for a whole genome sequence. I would actually go and pay for ctDNA and make sure I had minimal residual disease surveillance through my treatment, because I know that would give me the best chance. So there's the pressure from, that's what I see. That's what I see out there. That's what I see happening in trials and other areas. And the lady who spoke, Martina from Canada, before the session that I was on, she was saying exactly the same thing from Canada. She says, come on, let's get real. Let's get that ctDNA. Let's get the whole genome sequencing. Let's sequence the tumours. Because there's information there that we already know is going to help you with your diagnosis. There are so many aspects where if I was diagnosed 3C, I was that far away from being a stage 4. And once it starts spreading, it's really important to know all your biomarkers, not just the 2 that I was tested for, because it makes all the difference to what treatment's going to work for you that we know now.
But I guess the good news is from what you heard earlier, I mean, this GSK is definitely actively investing in the ctDNA space, and there's active research ongoing in the space by us and there's probably other competitors. So, there is a lot of active assessment. It just, I guess, from a patient perspective, the results take some time to generate, right? Because you're looking at longitudinal data and outcomes data. So I think I hear the frustration. You want to see that data immediately, and I can say we are certainly actively working on generating those data sets, but there's a regulatory ask to have robust enough data to make sure that there's also that you don't get incorrect news, right? That you either feel assured of safety, right, when in fact that there is actually active disease ongoing. There's obviously a risk associated with using relying on those types of products as well. So we want to make sure we get those right as well. So I sense the excitement, but we also want to make sure that the data is sound.
But it's there on that already now. So, for instance, colorectal tumors aren't typically tested for HER2, but like they are for breast cancer. But we do know that HER2 positive is a significant player in about, well, we don't know how many, what percentage of colorectal cancer, and of course, it's not tested for. Whereas we know it's at least 8%, it may be as much as 50%, but we don't test for it, therefore we don't know. And that makes all the difference to how you treat it. And repurposing of drugs like HER2 may be a progress.
Yeah.
There's lots of HER2 work out there.
Yeah, true.
Yeah.
So these are, that information is out there now, and that would benefit the patient to have that specimen.
Look, I need to call a formal close because we're already 2 minutes out of time, and the reception. But if you want to keep chatting, that's okay by me. But whoever wants to go, please do. But maybe we'll just take this one last comment.
Fully
if that's
agree,
all right with you guys.
fully agree with you. I think we should. The hopeful thing, very short, is that I think that maybe in 5 years' time, we will evolve to whole exome, whole genome testing anyway of tumor normal pairs for most of the cancers. And maybe when the monopoly of Illumina is finally broken, new players, then we will really see the sequencing cost drop. And once we don't have this expensive panel or library prep anymore, this will be a game changer. Then we will do a whole genome for like 600 euros. Yeah. Because today many people reproduce the slide that the sequencing cost is dropping all of the time. This is bullshit. I mean, in the last 5 years with indexation, we see the sequencing cost always increasing, panels becoming bigger. But I think new kits are, some patents are expired from Illumina. So there is some new companies coming on. So hopefully we have also the nanopore technology. So I think we will see some movement in that space. And as soon as as whole genome sequencing of a tumor or a tumor normal pair is the norm, then we come to an end of the technological development of the sequencing. And then we can really share really standardized data and go all in on the data analysis and data pooling. So then it will really kick off the Moon project for beating cancer.
All right. Exciting stuff. Any last comments from the panel?
Just one observation. In terms of technology, 2015 was a very long time ago. And technology and science, and we're just hurtling along. Unfortunately, we're working in a very conservative space.
Yes.
And we have to find ways to get rid of some of those anchors. And, you know, we remove some of those blockages because the technology is at that point of delivering the change that we all really want in treatments. But these are questions that go beyond science. They are political, they are policy, but that I can feel and you can see that the change will happen. And as you say, I don't know what will happen in 5 years, but we can only hope that the situation improves. And Ukraine.
Sure.
Okay. Thank you. On that
Thank you.
positive note,
Absolutely.
Thank you.
let's move on. Thank you very much.